Hi everyone, welcome back to Module 3, working with fMRI data. This is going to be the last video for Section 2, Intro to fMRI Data, and the topic is fMRI dataset processing and visualization, kind of covering Lesson 2 and Lesson 3 at the same time in this one video because the majority of the content for these two sections is actually going to be in the Google Colabs, and this video is more going to be talking about the context of the fMRI dataset that we're going to be working with. So what we know, first of all, from the last lesson, fMRI data is typically a 4D array data consisting of 3D space plus time as the fourth dimension. And fMRI data can be in many file formats, usually DICOM or NIfTY, and it can be loaded using the NI-Babel package. Once it's loaded, fMRI data can be accessed by the NIfTY object attributes. And then lastly, transposing the data is an important pre-processing step and a actually simple manipulation that makes working with the data a lot easier. So like I mentioned before, the key questions I'm going to answer here in this video specifically are what is the context of the experimental data set we're going to be working with in Lesson 2 and 3, and also what are some important tools for visualizing fMRI data, which I'll kind of go more into when I review or I guess preview the Google Colabs for you. So let's start off with the dataset context. So in Lessons 2 and 3, we're going to be diving into a real dataset a little bit. I've kind of extracted a little bit of data from this dataset just to give you guys a chance to work through some actual data. But basically, the dataset I chose is from openneuro.org, and this is a free platform with a lot of public brain imaging datasets. So if you're interested in looking for datasets, a really good resource to use. And the paper specifically that this dataset is from is called Neural Processing of Emotional, Musical, and Non-Musical Stimuli in Depression. So basically, this dataset uses fMRI in combination with an emotional, music, and non-musical auditory paradigm to kind of examine how neural processing of emotional auditory stimuli is different in patients with depression. So in this study, they had 19 people with depression and 20, quote-unquote, never depressed control participants who listened to positive and negative emotional music and also non-musical stimuli during fMRI scanning. And this was kind of to explore how emotion regulation is determined in part by emotional biases in cognition. So typically, individuals who have never had depression show a positivity bias in their cognitive processes. This kind of means that people without depression usually allocate more resources and more of their attention and memory for positive versus negative information, while individuals with depression show an overall bias towards negative information and generally tend to allocate more resources and memory maybe towards negative stimuli. So like I mentioned before, this study used a emotional music versus non-music paradigm. And so basically their methods were 5 fMRI runs. So during 3 fMRI runs, the participants listened to alternating groups of positive music, negative music, and something called pure tones, which were used as a baseline to control for auditory stimulation. And it was neither musical nor non-musical. It was just supposed to be very emotionally neutral compared to the rest of the stimuli. So that was basically the control. So during 3 of the 5 functional runs, participants listened to alternating positive and negative music with the pure tones in between for the control. And then during the remaining 2 functional runs, the participants listened to alternating groups of positive and negative non-musical stimuli, so just like noises or sound, and pure tones in between. So 3 functional runs with like positive or negative emotional music, and then 2 functional runs with non-musical, non-emotional stimuli. So the results from this paper showed kind of different results in different areas of the brain. They were specifically looking at the ACC, which is the anterior cingulate cortex. And what they found was that, first of all, in the ventral ACC, never depressed participants exhibited greater activation to positive versus negative stimuli, kind of as expected. And then in the rostral part of the ACC here, never depressed participants showed a greater activation for positive information, once again, kind of as expected. And then depressive patients showed a greater activation for negative information relatively, as you can see here, and also here as well. And then in the dorsal ACC, the pattern of activation was kind of different with the different types of stimuli. The never depressed patients showed greater activation to music compared to non-musical stimuli. So as you can see here, this is signal change, by the way. So below the signal change is a negative signal change, so kind of like less activation. So never depressed patients in blue showed greater activation to music compared to non-music stimuli, which is negative here, while patients with the depressive disorder showed greater activation to the non-musical stimuli. Here you can see it's positive, while it's very, very negative for the not depressed patients or participants. And in particular, they showed the greatest response to the negative non-musical stimuli. So kind of in conclusion, the study showed that in healthy participants, so not depressed participants, positive auditory stimuli activated the reward processing areas of the brain and actually showed differential responsiveness to patients with depression based on the different emotional and also non-musical or musical stimuli. So it kind of suggests that people with depression may process emotional auditory stimuli differently than people without depression, kind of based on the type of stimulation and also the emotional content of that stimulation. And so this kind of is an interesting thing to think about, I guess. And it's also maybe a potential path for more effective and targeted treatments for depression, especially if positive music can be useful in retraining that ACC function to maybe train the brain to have more positive responses to emotional stimuli. And so the role of fMRI in this is that it really helped localize the effects of these different stimuli, and it basically provided insight into like the very distinct results from the different stimulus types, and show that there might be implications for different therapies or risk assessment for depression. So kind of just a summary of what I just went through, basically just a little overview of the data set that lessons 2 and 3 are from. And if you want, definitely, I've linked it in the Google Colabs. Definitely go read the paper on your own if you're interested, if you want to kind of do your own analysis. But basically what's next is complete the Google Colabs for lessons 2 and 3. And then section 3 will be some more working with fMRI data led by Avi. So now I'm kind of just going to do a quick overview of lessons 2 and 3. So as you can see here, we're starting to work with the fMRI data set from that paper. So basically, I'm going to share the data set with you on Google Drive. Once again, this is like a very, very small subset of the actual data set. It's just looking at one, or I think 2 patient runs, because otherwise the data set would be like huge and it would take up all the storage on your computer from personal experience. So I'm only giving you a tiny subset, but enough to just look at these images. And so fill in the code once again, like lesson 1, using some of the methods that you learned from that lesson to kind of look into what these images might look like. So as you can see here, the array data isn't all just zeros anymore. This is actual experimental data. And you can look at the header again, and then we'll do the transposing that we need. And then we're going to talk about masking. And masking is a really important technique for visualizing fMRI data because As you'll see here, it's an important pre-processing step to get rid of unwanted noise and unwanted voxels and stuff like that. So as you can see here, there are a lot of data points around 0, which really isn't relevant. It's probably just noise that was picked up in the fMRI scans, and what you're actually interested in might be around here. So let's say above 700. So masking allows you to kind of filter out the noise and just focus on the important, like, I guess, or at least the parts that you're interested in terms of brain activity. So you'll kind of get a chance to work with that and mask and run this function, which, you don't really need to do any changes down here. Now it's more just exploring. But there's this cool function from the data connector, like Cognero course that I found. And it basically, once you run it, it allowed you to kind of slide through the brain slices and have like an interactive view of the data gathered from this study. And so I don't know if these sliders are working right now because I haven't run these functions, but you'll be able to do that on your own. And then lesson 3, which Avi made, thank you, is also going to be just another visualization of this data. You don't need to do any coding, just kind of go through it, learn a little bit about time series, and then we'll kind of do or show you more of a color version of these slices of the brain. And this one shows you, rather than the sliding picture, just all of them at once, all the different slices from this data. And so it kind of, we wanted to show you there's a lot of different ways to visualize this data, and also introduce the techniques used to visualize this data. And so once again, if you aren't comfortable with this stuff, just feel free to just run the cells and kind of just have fun exploring the different visualizations. But if you are, definitely like add cells, like maybe write some functions, see if you can do anything cool with these images, and just kind of explore them further if you want. And yeah, that's pretty much it for these lessons. Just use Google Colabs. Section 3 will be also very Google Colab heavy. So definitely let us know if you have any questions. And yeah, thanks for watching.